Now, as I told you already in the previous slide, that how the microsporophylls produce microspores because they hold microsporangia, which we called as sori in pteridophytes, and megasporophylls will hold megasporangia, produces megaspores. In the same way, in the present concept video, I am going to say you how the microsporophyll evolved to become stamen and anther in angiosperms, and how megasporophyll holding the ovules became carpel in angiosperms. This is a nice picture and good depiction. In this first case, I see a leaf. Naturally, what is this to me? This is microsporophyll. And what is it holding? It is lodging 4 microsporangium. 4 microsporangium. And what do they produce by meiosis? They produce microspores or pollen grains. Now, if you see in the next stage of development, the microsporophyll has become narrower and smaller in surface area. That means it is bringing the microsporangia together. So you can see in the cross section I have 4 microsporangia. So when the microsporophyll is curling inside, the distance between them is reducing. And if you see, this one stage is stamen. In case of anther, the microsporangia are clustered from the tip to their basal part. A complete, if I take the cross section which you are familiar in your textbook, it looks like this. So no leaf at all, no microsporophyll. That itself evolved to become a structure which held 4 microsporangia, the condition of tetrasporangiate in anther, which is a part of stamen. Now what do anthers have? Microspores or pollen grains. Then pollen grains are dispersed for pollination. All of you know this. So I need a holder which will elevate the anther for better dispersal of pollen to the pollinating agent. So we have a filament. So the entire stamen has evolved to have 2 parts, anther and filament, and this became the male reproductive organ in androecium of a flower. Androecium of a flower. I hope now you understood how we got the anther holding 4 microsporangia producing spores is held high by means of a filament forming androecium from its precursor, which has microsporophylls holding microsporangia. The same story with megasporangium. Again I have megasporophyll. So what are megasporangia called? Ovules. Now this also starts modifying by curling inside. Then the distance between ovules is reduced here, still reduced, and wherever I have ovules, the leaf gave a room and the other part of the leaf blade modified to become a passage which would allow the male gametes in pollen grain for fertilization, internal fertilization, and the tip of the leaf is modified to a receptive center stigma for the pollen grains which carry male gametes. So this one formed, as the picture shows, syncarpous condition all the carpels are fused, and it shows closely arranged 4 ovules. This is how it formed gynoecium. When we read morphology also we read the same. What is the female reproductive part of a flower? Gynoecium. What are the units of gynoecium? Carpels. What does carpel have? Stigma, style, ovary. What is ovary? The place where we have ovules. What are ovules? Megasporangium. What is the male reproductive part of the flower? Androecium. What are the units? Stamens. What does stamen have? Anther and filament. What is an anther? Microsporangia. Back to microsporophylls. Did you understand how the evolution from microsporophylls to anther and megasporophylls to carpels occurred in angiosperms?